So, very good afternoon, everybody. Now we discuss the today that is the another unit that the fungal diseases. So fungal diseases in two we have to discuss here. That yesterday we have discussed the rice blast disease and the remaining. Uh, this is white rust and wheat rust. We have to discuss rust disease. We have to discuss. So today we discuss the fungi produce the large number of plant pathogen and are responsible for the fungal diseases. Okay, so that is the major issues. Okay, few minutes wait. So these fungal diseases are the first important because they sometimes cause much damage and destruction to the plants by killing the cells and causing the plant stress. So here we can discuss the here sorghum, smart diseases and downy mildew diseases, bajra and powder we are to discuss. So smart of sorghum means jowar. Jowar is caused by the soil-borne faculty to biotrophic, that is the uh, Sphacilotheca. Sphacilotheca. So these three species of genus attack jowar plants, causing the very serious disease. So grains smut or covered smut of jowar is caused by the sorghum, that is the Sphacilotheca sorghei. Sphacilotheca sorghei. The last smut of jowar is caused by the that is the Sphacilotheca cruenta and Sphacilotheca raeliana is head smut of jowar. These diseases are very common in the state of Andhra Pradesh, Madhya Pradesh, Karnataka, and southern states of Tamil Nadu, Uttar Pradesh. These also. So symptoms of smut caused by the Sphacilotheca. The affected plants are smutted, produce thinner stalks, more tillers, and earlier flowering than the healthy plants. The fungus attacks the ovaries and all the ovaries in the head are turned into sore of the smut. The normal gain thus the replaced by an overall or cylindrical dirty gray sac with a slightly conical tip. The infected ear appear like a leafy structure. In most cases, stamens are absent, but the stigma is not transformed. However, other floral part of the host are not affected. So that is the mycelium is mostly intercellular without the astoria. The spirally branched, septate, and hyaline. The primary mycelium formed by the germination of basidiospores enter the host through the seedlings. When the primary mycelium of the opposite strain also happens to enter to the same seedling, the dikaryotic mycelium is formed through the anastomosis and fusion. This dikaryotic mycelium grows with the seedlings and produce the smart sore in the ovaries. So here diseased cycles. What here we can see the, so this is the, wait, you can show to another. This is also. So here the sorus means the grain of sorus, and this is the structure of the different. So all time of the flowering or diploid phase leads to the production of teltospores, teliospores. They are replaced as seedling production in the host. Heliospores over winter in the soil, and crop debris provided primary inoculum for the next cycle of the infection. So when such a, so here the teleospore germinate along with the seed by first forming a thick, usually four-celled promycelium bearing the lateral sporidium. The sporidium are oblong with the slightly rounded ends and spindle-shaped. The sporidium germinate and infected the developing sorghum seedling. Sometimes the teleospores germinate directly by the producing germ tube. The promycelium may also act as the infection hyphae with the may enter the ceiling of the host. Generally, the sporidia fuse in the pair of the from dikaryotic mycelium. The dikaryotic mycelium enter into the seedlings. So here the dikaryotic mycelium enter into the seedling. This diagram see here. This one is the mycelium. So mycelium structure is the. Uh, so most infected here. That is the pH temperature here 6.68 to 78 Fahrenheit. That is the, and that is the grain smart. The grain smart also smart. So grain smart is also known as the kernel smart. So this covered smart or short smart is covered by the fungus Stachylotheca sorgi. That is a sorghum is there. So that's a Stachylotheca sorgi. So cover the kernel smart is generally distributed in every sorghum growing region of the worldwide. Major construction constraints to yield of the small holder sorghum is semi-arid areas. So symptomology is important. The individual grain is replaced by the smut sore. The sore are overall all cylindrical, covered with the uh, top cream skin, which is the often persistent, unbroken up to harshing. Ratoon crop exhibits higher incidence of the disease. So the seedling plants are infected, but symptoms generally are not apparent until booting or heading. The teleospores are often fungus replace the gain in the panicle, causing the direct crop losses in the promotion to the area of the panically infected. This is the infected uh, Saurus. So head smart and so head smart is a disease by the fungus that is the Specialistica riliana. So head smart is important to sorghum disease because it is a presence in all part of the world where the sorghum is grown in the potential to the cause yield losses and due to the natural variability. Soil-borne smart spores from the previous crops can be infectly young sorghum seedlings, which may retain the relatively healthy in the appearances. So that the symptoms that appear at the booting stage when the entire head is replaced by the large sore. The sorus is covered by within the a gray membrane of fungal tissue, which is before the head enter into the fungus boost leaf to expose the a mass of the brown or black smuts. Okay. And so from the here the head may be totally smart with the characteristic witches broom is many small rolled leaves producing the head of the sucker at the nodes. So Specifica rhelium is one of the few smart that is cannot be controlled by the seed treatment. Living host to resistance as the only the practical control mechanism. So loose smarts. So the disease caused by the fungus Specifica creanta is called as a loose smart or kernel smart, which is the less widespread the covered kernel smart. So symptoms, what are the symptoms? The affected plant can be detected before the ears came out. There are the shorter than the healthy plants with the thinner stalks and abundant side branches. The ears came out the much earlier than the healthy ones. All the spicklets of the infected ear are malformed and hypothetical. The sorus replaces the pistil and stamens and is borne on glumes and pedicel, covered by a thin membrane with a rupture very early, exposing the spores even the head emerging the sheath. The affected ear head appear like a leafy structure. So see here that I shown to that diagram also, which I show that picture also. See here. So this one is the some smart diseases. So management for all smarts. The treat seed or copper or captain or therium at 4 grams per kg. So this is free seeds we can use it and follow up the crop rotation and collect the smart earbuds in cloth bags and bury in the soil. When the possible, promptly removing and burning head smart is remembered to prevent the skating the spores. So the green ear diseases are downy mildew of bajra. So bajra, see here, this one is the bajra. Uh, so this is the bajra. So green ear disease or downy mildew of bajra is commonly disease occurring in all the countries where the bajra crop in grows. In India, the disease was first reported and studied by the battler. In India, downy mildew epidermis caused the sustained yield losses during in 1970s. So symptoms on seedlings and inflorescence, these are there. The infected seedlings turned pale yellow and became a stunted due to the shortening of internodes. The infection at the in early stages cause the stunning with the any Tyler formation. Infection at the latter, the causes stunning with the excessive uh, tiller formation and thus the plant became a bushy. The leaves of the infected plant develop pale and chronic, that is the bread restriction on the upper surface and on the lower side of the leaves. Some that serve infection entire the foliage became the white due to downy grows on the fungus and can be seen on the upper as well as the lower surface of the leaves. So if the inflorescence also affected by these fungus, or the infected plants fail on the form the earbud or form the different floral pot including the glumers and stamens, pistils, etc. They covered into the green liner leafy structure of the different green ear diseases as the disease advances in the malformed floral structure of the ears became brown and dry. So here this is the Bajra disease. So sexual reproduction here, oospores are formed by the antheridium and oogonium. The oospore are oval or three-walled and 39 to here millimicrons and develop in the large number inside the mesophyll. The oospore the penetrate for the long and period uh, during the unfavorable conditions and with the one set of favorable conditions. They are germinate by developing 1 to 4 germ tubes. Disease cycle. So disease cycle is a systematic. The pathogen is obligate parasite and primarily soil borne. The pathogen survive in the soil or primary infection is caused by the oospore. With the insect of the favorable condition, the oospore germinate and enter the young seedlings of the host to systemically through the underground part of the and cause the primary infection. So how we have to control these? Since the pathogen is primarily solid borne and persistent in soil up to 5 years, so crop rotation is most important and non-host type. So that is the removal of diseased plants and burning immediately. Check the disease again if the treatment is occurred. And seed treatment here agrogene and present their the use of the disease resistance varieties that H1B, HB1, HB15, HB10. These are the some variety disease varieties are there. So next one is the powder mildew of cucumber. Cucurbites means cucumber family members that cucurbits and shown to the diagram. This is the downy. Uh, so powder mildew is a common fungal disease, the worldwide occurrence on the cucurbit crops and other vegetables and pulses also. The first sign of the powder mildew, pale yellow spots and the leaves, stems and petiole. So here powder fungal growth develops on the both leaf surface, petiole and stems also. The large blotches gives on the lichen and powdery appearance. Leaves of the severely infected plants to turn brown and fall the permanently. When powder mildew infect the majority of the foliage, the plant weakens and the fruit repents. prematurely, thereby result to the reduced yield and market quality. So here causing organisms, that is the Podospira, Xanthi and Erysife, Sycoramciarum, Sycoracearum, that is the Erysife sycoracearum. These are the most common fungi causing the powder mildew of the cucurbits. So Pseudospira xanthil and Erysife sycoracearum. Erysife sycoracearum. The mycelium is well developed, specifically the well developed Astoria in the host cells. So here, so Clisthetiska are the globose scattered gregarious with a thick wall. The Clisthetiska are surrounded by numerous mycelial appendages about the uh, large times than the diameter. So acid are the ovate stacked 12 to 25 for Clisthothium. The ascosporium are two in each ascus. Two ascus are present. Two spores are there. So this is the cycle. To here, this is the cycle. So here, the source of initial inoculum for the powdery mildew have not been differently entered the primary inoculum in the airborne. So conidia disappeared potentially. The conidia remains for the 7 to 8 days. The casual agent is the obligate parasite and therefore cannot survive. So here the control, how we have to control these. So pruning or removing the diseased foliage from the plants and destroying that we can use the resistance varieties. Resistance varieties also we can use the and that is the biofungicides we can use and affected fungicides finely powdered sulfur and lime sulfur potassium bicarbonate we can use it to control these diseases. So next one is the downy mildew on grapes. So here downy mildew on the grapes. I can show the diagram. Uh, Downy mildew on. So this is the downy mildew on grapes. This is the grapes. So downy mildew of the grapes. Right. So downy mildew is a wide spread or highly destructive disease of grapevine in all grape growing areas of the world that can result in severe crop losses. So early infections of young uh, bunches can lead to significant crop loss, where the sour leaf infection may lead to the defoliation and possible sunburn and lack of the fruit ripening. So the pathogen here, Plasmospora viticola. So Plasmopara viticola infected the above ground part of the grapes vine, especially the leaves. The pathogen infect all the tender plant pods, initially showing round, light green spots of an oily appearance on the upper surface due to the increase in size and spots influence to the cover most of the leaf surface. As the disease progress, white cottony grow, and the disease gets to its name to downy mildew. From the presence of downy growth, the several infected leaves will dry up to fall permanently. The infected shoot tips became thick and curly lateral bore turn brown and die. The young shoots became the dark red and dandruff. Mm, the fruits and fruit rot and branches fall in the ground. It is infection occur only the young bunches stalks to enter the inflorescence may die, destroying the entire crops also. So this is the fragile organisms, that is the plasma plasmapara viticola. This is the infected cycle. I show to the infected cycle also. Wait, this is the infected. So this is the cycle. The sporangia found on the leaves The leaf may be remain on the veins all years around. In such cases, the infection by sporangia may be continuous from the zoospores are formed during the oospore germination as are covered by uh, the pushing raindrop from the soils on the lowermost leaves of the veins. So here the pathogen takes place through the stomata. The mycelium invades the intercellular space. The ramifies in the host tissues, deriving the nutrition. That is the long measure through the Astoria, which penetrate the cell lumen. In almost all cases, infection of the leaves takes that is through the lower surface only. So this is the life cycle here. So how the controller here sanitation is most important. Destruction of the old leaves to eradicate the surface, the source of inoculum. The all green shoots or the sucker that the develop at the base of the uh, stalks should be removed as they very susceptible to primary infection. Borax mixture that is the important here. Borax mixture also. So we can spray this Borax mixture to remove the epiphytic condition. This is where the disease can be affected effectively controlled by the spraying the vines 30% of Blatox and diethyl Felban or Captan. Okay. So next is the corn smut of maize. See here, that's that diagram also I shown. Wait, this is the carbon means corn. This one is the corn smut diseases. The corn smut disease caused by the Eustago maedis. Eustago maedis infect the maize and is furrow-wise distribution. In India, this is occur where the corn is growing, particularly in Punjab, UP, Kashmir, and which is reduction in the yield and almost to further and inferior kernel roster of the smut ear and the lower carbohydrate content. So this is the symptoms. So smut infection often result in the abnormal growth of the plant tissues into the tumor-like galls. The disease is easily to recognizable by the presence of large knob-like sooty, the swelling or boils known as before the formation of smut balls. Okay. The latter appear on the ears or stems. The gall may be appear on the stem leaves and axillary buds. The gall at an early stage are light colored and are the edible. At this stage, they are the covered with the firm shining membrane. So this is the see here um, corn smut. So this is the corn and spores are germinated and mycelium to develop. This is the very serious condition in the smart corn, smart and maize. This is the spores will develop. So here the disease cycle here. The fungus infected all parts of the host plant by the invading the various of its host. The primary inoculum come from the airborne and waterborne Sporidia and Basidiospores, which are produced by the teleospores. Over-iterating on the crop refuse in the soil, or rarely on the grain itself. The germination of teleospores. To germinate under the suitable condition, temperature and moisture, each grown into a septate and slender promycelium called the peribasidium. It is a four-celled structure. Each segment of the epibasidium bear over weight and unicellular hyaline spores or sporidium. The sporidia multiply by the budding in the yeast-like manner. So germination of the basidiospores and infection. The sporidia are dispersed by an air um, currents that water. They fall on the corn plants. They are the embryonic tissues of all the above ground parts are susceptible to direct penetration. Here, each basidiospore germinate by the producing a germ tube or the primary hypha. Indirectly penetrate the corn tissues, entering to the epidermis is growing the um, that is horizontally beneath into from the primary or monokaryotic mycelium. Active infection takes place only there are the two hyphae from the neighboring primary mycelia of the opposite strains come into the contact. So formation of the gall or tumors. So that is the in the response to the fungus infection, the host cells are infected region and the size here, the number and consequently the tumor outgrowth develops. It is permanented with the secondary mycelium. The fungal hyphae at this stage became the additionally septate into smaller binucleate segments. These segments round off and separate into binucleate smut spores. So meanwhile, the host tissues in the tumor expecting the superficial layer and disintegrates. The tumor is now filled with the mass of myriad of black smut spores covering the membrane trace and finally rupture to release the smut spores. They are the blown about by wind or dispersed. Eventually, they return to the soil and remain the dominant in winter. So here disease control, how the disease control, that is the rotation of the crop varieties, rotation of crops, and sanitation assistance is reduced to damage, and collecting the smut tumor from the field and before smut spores are released and burning them to use to control measures. The infection is incidence can also be reduced by the application of insecticides such as the DTD or nicotine we can use. So groundnut leaf spots that we call as the ticka disease. So the disease here I show to the ticka spots, ticka disease the spots show. So this is the ticka disease. So black spots are appear on the leaf surface. So this is caused by the cercospora and known as the cercospora leaf spot or truly known as the ticka disease. So it is the serious disease in the ground growing area of India and cause the about 25 to 50 reduced in yield. The size will be quality of the nuts is generally affected. So symptoms: all the area spots are post plant attacked by the pathogen, shows characteristic symptoms and limitation. Initially, the lower leaflet get the infected, showing dark spots, and later can each spot became surrounded by the yellowish halo. The large number of spots, almost circular in out, develops on the leaf. This is the smut leaf, means ticka disease the leaf. The stem and petiole also spots, but the less in number. With this, every the disease and they became calculated and defoliation of the leaf takes place due to the defoliation, the size and quality of the nuts became generally reduced. The infection at a younger stage causes defoliation and nuts fails to develop into the that is the Cercospora personata and the Cercospora arachnicola. These are the two species are there. The casual organisms, Cercospora personata and Cercospora arachnicola. These two genus fungus species. They are two distinct are there. So another diagram I show to this one. So the this is the of the groundnut. Conidia sardine. So pathogen can penetrate through the conidia or diseased plant debris remain in the soil, on the shell, the fruits and also on the seeds. The infection is forwarded in high temperature ranges between 25 to 31 degrees and high humidity and continuous low temperature along with the while also the fever infection. The infection takes place through the stomata or by the accessing the host to epidermal cells. So this is the control here. There are the cultural method and sanitation, cleaning, the destruction of burning planted debris. Crop rotation is also early showing. This one is to early showing is also most important because control the diseases and use the resistant varieties also. So here chemical method is we can see use the seed treatment, that the seed treatment with the using the aggression and foliar spray borodox mixture and systematic fungicide that is a biovestin and a belton. Okay. So next one is the cotton wilt disease. So cotton wilt is the globally important crop used for the both the natural fiber and seeds. So fusarium with the So symptoms is affected at the all stages of plant growth. In the seedling stage, there are the yielding, yellowing to cartelians, growing of petiole, followed by the wilting and drying the seedlings. In some young and so growing the blackening of wells. The seasons are stunted with the fever boiled with very small or opened before they are the mature. So here fusarium with the symptoms I can show. So fusarium, another one is the fusarium oxysporium and fusarium versicolor infection. So this one is there. So another we have to discuss that is the bacterial and viral and mycoplasma diseases. Bacteria, virus, and some mycoplasma diseases. So the bacteria are the microscopic single cell. There is, so here the bacteria are microscopic, slightly celled that prokaryotic organism. The bacteria cause many diseases in the plants. The symptoms were that causes the bacterial infections can be necrosis, hypoplasma, um, hypertonic, and so the mycoplasma here that is the bacteria related to gram positive. There are the simplest and smallest known organism. So bacterial disease that is the necrosis. So the death or killing of the host tissues induced by the attack the pathogen is called as a necrosis. So here the bacterium or fungus that is rotting the universal plant rods and many kinds. The common ones are soft root and dry rot and where rot is hard before the soft rot attack large number of hosts and best known causing the disease that is the fleshy plant. So canker, another one is also canker. Here lesions, canker are pocky outgrowth formed and trunks, stems, twigs, and branches also, other ground plant parts also. Canker can be slightly sunken, dark brown, and large amount of. Next one is the tumors or galls forms. So tumors also here formed the like here you can see. So this is the some bacteria and fungal diseases, and scapes. The bacteria scapes plant primarily infected below the ground parts of the plant. That is the common scapes are caused by the small number of that is the streptomyces species with the caused the localized scabby lesions on the outer surface of the tuber. Typically, the Hurki tissues will form the below and around the lesion. Rat pathogens can gain the entrance into the host tissues through these lesions and further degrade the host. The vascular diseases means the vascular wilt. So this one also, and another one is the paddy blight disease. So paddy blight, that is the bacterial blight, that one we call. So bacterial blight.